Good morning, everyone. I'd like to thank the Pescola Foundation and the ACR for the privilege of selecting me to receive this award. I'm grateful to the foundation and the ACR and the president of the ACR for this opportunity. Thank you for this introduction. I'm pleased to be presenting and pre-recording this talk for the ACR Virtual Symposium 2020. For the last 10 years, I've been a group leader at the Cancer Research UK Cambridge Institute, part of the University of Cambridge. It's based on the Cambridge Biomedical Campus near the Addenbrooke's Hospital, and it's a fantastic place to undertake both basic research and translational research, which has been our focus for the last 10 years. I'd also like to particularly thank my colleagues and the staff and my team members, and especially the directors of the Institute over the years who have given me the opportunity, particularly Professor Sir Bruce Ponder, who took a chance and allowed me to open up my lab and to set up our research group focusing on circulating tumor DNA. And for the last 5-6 years, I've also been CSO and co-founder of Innovata, a company which we spun out from work, some which I'll tell you about today. And I'll show you some of that as we go through the talk today. So I'm going to introduce some of the background and some of our early work, especially in monitoring circulating tumor DNA. Then I'll tell you about how we've developed and used targeted sequencing to study circulating tumor DNA and how we've taken that through into clinical implementation. The main part of the talk will focus on sensitive assays that we've developed to study residual disease through circulating tumor DNA. And the last part would be a brief sneak preview of new data that's coming out from our lab just now. The objective of our study is to understand and study and develop diagnostic tools for cancer. And cancer has its own natural course. It may start as a small tumor. At some point, we may pick it up either through symptoms or through various screening modalities. Then we try to treat cancer. After treatment, either cancer has disappeared from the body, or if a small amount of the tumor remains, it may develop to metastatic tumors, which may then vary or differ from the original tumor. And through this course of disease, we have many opportunities to help in the management and treatment of patients by providing better tools to treat cancer, from detection of cancer in its early stages, through molecular profiling or stratification to help decide the best treatment for cancer, through monitoring and detecting residual disease and early detection of relapse if the patients have been treated. And at the point of advanced cancers, we can also study tumor evolution through these means. There are different ways that we can generate diagnostic information to help guide treatment. Imaging is an important tool for understanding and diagnosing cancer. It's a non-invasive tool, so it can be done repeatedly, but it provides limited molecular information. So it gives us a sort of black or white, or in some cases, a grayscale picture of cancer over time. Biopsies are an important way for us to obtain tissue material, and they're part of the diagnostic process. However, they're invasive and sometimes expensive, so they only provide tumor material in limited time points and also limited sampling over space if there are multiple tumors in the body. Over many decades, blood-based biomarkers have been developed and studied because these can be minimally invasive, and if they can work, they can provide a lot of information. However, 10 years ago when I was starting this work, there were few available blood-based biomarkers that had sufficient specificity to know that the signal is coming from the tumor. Therefore, the amount of information that we could extract on the cancer from blood biomarkers was limited. And in that setting, we started our work studying circulating tumor DNA, in a large part inspired by work going on in parallel fields like NIPT, led by Dennis Lowe and other colleagues. And to just show you how the field has developed over these years, when I started my lab in late 2009, it was possible for someone to actually go through and read each and every paper ever published on cell-free DNA or circulating tumor DNA. And the term liquid biopsy was not yet used. It was introduced by Klaus Pantel sometime after this. And if you look today, actually, there's almost 1,000 papers already published on cell-free DNA, circulating tumor DNA, liquid biopsy from the start of 2020. So it's no longer possible for me to try to ask my students to read all the papers or even all the papers published this year. And it's been a privilege to be part of this progress in the field, and great fun too. Some brief introduction onto what we know about the biology of circulating tumor DNA, although this is not the focus of the work in my group. Tumor DNA arrives in the circulation through various cell death mechanisms such as apoptosis, necrosis, and there are speculation that it may arrive through active release through exosomes or other processes. And when we then go and look for the DNA in the blood, we can find point mutations, we can try to detect copy number alterations, rearrangements. Importantly, in the recent years, is also the ability to study methylation changes in the ctDNA. There are challenges in the study of circulating tumor DNA, and I like to view them in these categories. One is that there's very small amounts that we're working with. So in a blood tube, you can obtain, after you spin down and remove the blood cells, the amount of cell-free DNA that you obtain in plasma is on the order of a few nanograms of DNA, so only a few thousand copies of the genome. Of this small amount of DNA, only a small amount may come from the tumor. In advanced cancers, this could be as much as 10% or more of the DNA, but as you go to earlier stage cancers or after treatment, this can be much below 1%, as I'll show you in the data to follow. And the last part is really the consideration of error rates, because from this small and limited material, we're trying to get as much information as we can. So we're using methods based on sequencing and PCR, and these introduce error rates, and it's important to keep these errors under control. A big part of the work in our group over the years has been the development of increasingly powerful tools to get more and more information out of circulating tumor DNA. And it can be used either as a sensitive way for tumor quantification in the blood, or if you go towards more wide-scale sequencing tools, it can be used for molecular profiling. And in our lab, we've worked with tools such as looking at individual molecules or mutations, to the other end looking at genome-wide analysis. But really, the main part of our work has focused on targeted sequencing and various flavors of targeted sequencing, which we found to be a very powerful tool for studying circulating tumor DNA. What the field has found over these past 15 years or so of intensive research in the area has been that we can demonstrate now very conclusively that the levels and the molecular composition of ctDNA reflect very well what's happening in the tumor. And this means that this opens up a wide range of possibilities for diagnostic applications. As I mentioned, in earlier stages, we can try to detect cancer or even develop screening tests, as has been now done in recent years. We can try to perform molecular profiling, we can monitor it, and so on. And we've been involved in studies in all these different areas. I'd like to start with introducing what for me was a big step in progress in this field, really a breakthrough to demonstrate that what we're detecting in plasma is specific to the cancer. And this is through the process of personalized cancer monitoring. And this was developed largely by a group at Johns Hopkins and led by Bert Vogelstein, Ken Kinsler, and others, and demonstrated very nicely in a set of 2 papers by Frank Deal, Luis Diaz, and others in PNAS 2005 and in Nature Medicine 2008. And this is the idea where you start with a tumor biopsy, you sequence the tumor biopsy to identify mutations that are specific to the tumor, and then you use these mutations as your markers to look and to quantify tumor DNA in the blood samples. And thereby what you detect in the plasma is you can develop tools that are sensitive, and it's both specific to the tumor. And when we started the lab in 2009, we followed this process, and I'll show you briefly some of the results that we've developed in these early years. So this first study is from a joint work we've undertaken with Carlos Caldas at our institute. And in this work led by Sarah Jane Dawson and Dana Tsui, we looked at a cohort of patients with advanced metastatic breast cancer. And through the collection of plasma samples and the parallel quantification of CTCs, we were able to show that although these can track well together, the number of cell-free tumor DNA molecules was much larger, about 100-fold more than CTCs as could be quantified with that point-of-Veridex system. And we could also identify the prognostic component or contribution of ctDNA. And what we could see that by comparing to CTCs, CA153, and imaging is that ctDNA provided the earliest indication that cancer was progressing in this cohort of advanced breast cancers. At the same time, we undertook another study looking at advanced ovarian cancer together with James Brenton. In a study led by Christine Parkinson and Davina Gale, we looked at a cohort of advanced ovarian cancer patients. And there we studied, we monitored ctDNA through individual mutations in p53, following the discovery a few years earlier of the very high prevalence of p53 mutations by James Brenton at our institute. So here we see the levels of p53 mutations normalized to their level pre-treatment. And you can see here that these drop very sharply after starting treatment for these advanced ovarian cancer patients. And you can compare that to CA125, which is the acceptable, the clinically used marker. And you can see that the response is much faster for ctDNA. And if we separate the patients into those who respond and have a big drop in ctDNA, and those who do not, you see that the prognosis is much better for patients where ctDNA levels drop very quickly. One of the other observations we made in these studies of advanced cancer patients is that in some cases, the levels can be really shockingly high in these patients. So you can see here, for example, one patient from this ovarian cancer cohort, where pre-treatment, 20% of the DNA in the cell-free DNA in the plasma had this mutant p53 allele. And after additional rounds of chemotherapy and response and then progression, even in this point, almost 50% of all the p53 alleles in cell-free DNA carried this tumor-specific mutation. And one of the ideas that we had in the lab, and this was work led by Mohammed Murtaza, who was then a PhD student in the lab, was that if we take these samples with very high levels of ctDNA, we can actually study these by very wide-scale sequencing. And he proceeded to very rapidly demonstrate that we can perform exome sequencing on these time points. And by comparing the pattern of mutations before treatment and after treatment, we could generate information that can help discover potentially novel resistance mechanisms. And this paradigm has now been followed by many groups studying response to novel therapies and possible resistance mechanisms. So having shown and demonstrated that ctDNA could be a good way to understand what's happening in the tumor, really wanted to see how we can get more information out of this and how we could potentially develop this as a practical tool. To do this, Tim Forshu, who was a postdoc in the lab, developed together with others in the group this method for tagged amplicon deep sequencing. And to my knowledge, this is the first method that has been developed for targeted deep sequencing of cell-free DNA as a liquid biopsy. And we did this by separating and identifying the regions we want to study in the genome and capturing these by a series of short overlapping amplicons. And the amplicons need to be short because the fragments of DNA in the plasma are short. They're on the approximately on the order of 150 bases, one nucleosome size. And then this PCR process was built and developed into starting with a preamplification process, further cleanup process by singleplex PCR, barcoding, and then deep sequencing. And this allowed us to generate data and to make several interesting observations. Here's one of the cases from this initial study, where we looked at a patient who had initially two concurrent cancers, one an ovarian cancer with a p53 mutation, and the other a bowel cancer with a different set of mutations, including a different p53 mutation. 5 years later, the patient had a cancer recurrence, and we were able, through analysis of the blood sample by the single multiplex assay, to tell which one of these cancers is the one that recurred. In another example from the study, we wanted to demonstrate that we can track multiple mutations in parallel together from the same patient. So in one breast cancer patient, we developed a personalized panel sequencing assay looking at 10 different mutations, and we could show that in this instance, they all tracked well together. We later developed that approach further by work led by Mohammed Murtaza. We looked at one of the breast cancer patients from the previous study I described, and by collecting multiple tumors at the time of autopsy, these were exome sequenced, and we then developed an assay to look at 200 different mutations that Murtaza tracked over time. And you can see here data for 200 different mutations in 9 time points in the plasma and in different parts of the tumor. And this allowed us to identify the particular subclone of the tumor that was progressing, and that eventually led to this patient's demise. But the thing that I got initially most excited about was this ability that we've developed to non-invasively profile the tumor for mutations. And we really were convinced by this through this first case, where we initially had sequencing data from the tumor where we found the p53 mutation. And when we applied our TAMP-seq platform to the plasma, we found this p53 mutation as well, but we also found an EGFR mutation. And this EGFR mutation was not found in the initial tumor. And then by looking at 8 different tumor specimens collected at the initial surgery, we did find evidence of this tumor at very low levels in one of the omental masses. But actually, a year and a half later and 2 years later, we could identify this directly in the plasma in a non-invasive way. So having come from the background of previously working in a biotech company, I thought that the way to progress this towards rapid clinical implementation would be to set up a spin-out company, which is what we did. And in 2014, together with James Brenton, Davina Gale, and Tim Forshu, we spun out a company called Inivata, which has its headquarters outside Cambridge and has a clinical lab in the US in RTP, North Carolina. And what we did through Inivata is we first developed further and improved this platform to what we call the Envision platform, described here in a set of papers by Davina Gale, Vincent Planiol, and others, where we took the deep sequencing platform, we improved the PCR process to reduce the noise, added improved analytics based on the replicate analysis, and enhanced our deep sequencing from 700x, which was deep in 2012, to 70,000x, which we do now. And through these processes, we're able to significantly improve the sensitivity of the assay. I'm showing here the analytical validation of this assay, looking at different allele frequencies and the sensitivity we obtained. And you can see here that even going down to allele frequencies of 1 part per thousand here, we still retain significant sensitivity here, about 80% and almost 90% at 0.2% of allele fraction, meaning two molecules per thousand. And you can compare this to other platforms that are being used today, and we feel really pleased by the sensitivity of our platform. And this is now applied on a panel of 37 genes that are particularly focused at helping guide treatment for non-small cell lung cancer, including all of the NCCN guidelines recommended mutations. This graph shows the time course for ctDNA for the first patient who was treated on the basis of information we generated at Inivata. And we received from our collaborators at the Institut Gustave Roussy outside Paris a blood sample from this patient collected in April 2015. And by profiling this with our platform, we identified a set of mutations, including, importantly, this EGFR T790M mutation, which made sense because this patient had previously been treated by EGFR TKI. On the basis of this information, they could obtain access to compassionate use of osimertinib, which was not yet licensed at the time. Patients started on osimertinib in June 2015, and by July, you could see this rapid response and a sharp decrease in levels of ctDNA. You can see here multiple mutations whose levels come up and down as patient had additional progression and additional treatments. Interestingly, the T790M mutation, which we identified initially, this mutation we never saw again, suggesting that the osimertinib actually did its job for this patient. Other effects you can see here, for example, different mutation in 53, which didn't play along, and maybe this represents an instance of clonal hematopoiesis, which at the time we didn't have the tools to study. We follow this further by a set of studies, by a study looking at a set of 48 patients who were prospectively treated on the basis of plasma-based analysis. To my knowledge, it's the first study where patients were prospectively treated in a cohort basis. And out of 48 patients who were treated with EGF-TKI, we found 50% of them to be positive for T790M, and these were subsequently treated by osimertinib, and we had a response rate of 62.5, which is analogous to the response rate you would obtain if you treat a cohort of patients who had T790M based on tissue profiling. This was followed further by additional clinical validation studies, both with the group at Gustave Roussy, by Jordi Ramon, Benjamin Bess, and Jean-Charles Soria, and led by a group in the US by Michael Pritchett, Ramaswamy Govindan, and others. And across these two studies, we could demonstrate high concordance and high sensitivity of ctDNA when compared to tissue testing. Interestingly, even though this study had fully paid for tissue testing in the US, because of the difficulty of accessing tissue as part of routine clinical care, we ended up having more mutations where actionable, more cases where actionable mutations were identified in plasma compared to the tissue. And on the basis of this and additional data, the assay was received a local coverage decision in the US, and it's now covered by Medicare for patients in the US. I'd like to focus, though, the rest of the talk on our work that we've been working in parallel to try to push the boundaries of sensitivity to help us tackle this really challenging problem of detecting residual disease. And the problem we aim to address is this, that after treatment with curative intent for early stage cancer, patients, even if they have no signs left of cancer, are still at risk of having relapse. And many patients do receive adjuvant treatment, even though the vast majority of patients do not benefit from this treatment. Only a small minority, a few percent, benefit from this treatment, but all of them are subject to the risks of the side effects. So if we could identify better which patients are the ones at risk, we could do a lot to reduce this burden of side effects, and maybe even identify patients where we could give more aggressive treatment if that's necessary. We could also help streamline and expedite clinical trials, because at the moment clinical trials need to be very large in this setting because of the very small effect, because most patients are already cured. That's the good news. So the ambition here is whether we could take a blood sample, and from a blood sample, taking this time point, we could differentiate the patients who have some signs of residual disease, and maybe these are the ones destined to have a relapse if they're not treated, from the ones who have already been cured, or at least we don't see any signs showing a good prognosis. And once we have this assay and this technology, even if it's negative in the initial time point, we can continue. And even in cases where we couldn't detect, we might be able to detect the occurrence of relapse earlier than we would otherwise detect it through other means, such as imaging. And we were encouraged and highly motivated to follow this path through two things. One was the early emerging evidence showing the potential of this, even through assays that were somewhat limited in their sensitivity. I'm showing you here one example and then another example from two studies which only looked for a single individual mutation in plasma and tried to differentiate patients who had one mutation versus patients where one mutation was not found. And you can see here in this study in early stage colon cancer by Johns Hopkins and a group in Melbourne led by Jeannie Tai, you can see here patients who had ctDNA detected had a poor prognosis and relapsed quite quickly. In contrast to patients where they did not detect ctDNA, had a much better prognosis, although still had significant occurrences of relapses. And likewise, here in this study in early stage breast cancer by Nick Turner and his group, Isaac Garcia-Morillas and others, where they looked at early stage breast cancer after surgery. If they had ctDNA, these patients relapsed within a year, and the patients who did not had a much better prognosis. But still, this prognosis was not good enough to tell people that they might forego, for example, adjuvant treatment. So the ambition here is to have a more sensitive assay so we could identify more of these patients, give these adequate treatment, and maybe be able, in a position to advise some of these patients to take less aggressive treatment or no treatment. And the way we wanted to do that, us and others working in the field, is to multiplex our assay. So instead of looking for a single molecule or a single needle in a haystack, we now want to look for as many needles as we can to try to improve our ability to do that. And so that's where the second piece of the motivation came in, because we already knew that we can do this quite effectively from our study in 2015, where we showed that we can monitor hundreds of mutations in parallel. So if we could scale this up and even increase the numbers, we could push the sensitivity even better and not look at individual clones, but even at recurrence of the tumor in general. So several groups have been following this path, and there's been a lot of exciting evidence generated over these 5 years. One of the notable examples is work from the TRACER-X group by Charlie Swanton, his paper by Chris Abosh and others, where they used a technology by by Natera, where they looked at a set of about 20 mutations that were previously identified in the tumor. They monitored these in serial blood samples, and they found that indeed they could have a lead time and detection of ctDNA before relapse, and about 77 days lead time in this cohort of early stage non-small cell lung cancer patients. Other groups have done this and further. Oh, sorry, I wanted to mention also that in the recent ACR, first part of the ACR, we also saw some further work developed through this work, and there's more data coming out. Other groups have followed and have also looked at other ways to look at even more mutations. Multiplex and his group in the US and TGen have looked at a cohort of patients with early stage breast cancer, and through an assay they've developed, they also looked at the ability to identify patients who had pathologically complete response after neoadjuvant treatment. And also in breast cancer, a group at the Broad and Dana-Farber looked at also breast cancer, and they found that patients where they could track more mutations in the plasma had a better sensitivity and a better chance to detect MRD. Also in recent exciting work by Max Dean and the group in Stanford, they found that patients with early stage lung cancer, where they could not detect ctDNA using a population-based assay, by taking a patient-specific assay looking at many mutations, they could now go back and pick up these mutations. The presence of ctDNA in the plasma. So the rationale for this is really this, that if you have a blood sample and it has a limited number of copies of the genome, if you look for an individual locus, your sensitivity is going to really be limited by the number of genomes in your sample. So it doesn't help if your assay has a analytical sensitivity of 1 in 100,000. If you only have 5,000 molecules, you're not going to detect lower levels than 1 in 5,000. So one way to do that is to try to collect more material by collecting more blood or doing plasmapheresis or other ways to enrich. But the other way is to expand this way and to look at multiple mutations. And as I said, various groups have been looking at whether they can detect any number of different mutations here. In order to do that, you first need to know what are the mutations you're looking for. So usually these approaches start by undertaking some form of tumor sequencing, identifying these mutations, and then looking across these loci. And then oftentimes what these groups try to do is they try to call each of these mutations. So they have n times shots at the apple, but each one of these they're trying to call a mutation. What we wanted to do is something slightly different, where instead of calling individual mutations, we group all of these molecules together, and thereby we have many more molecules to look at, but that requires a different analytics. But that could push our sensitivity down to really few molecules in the whole number of molecules we're looking for, which could be very large, as I'll show you shortly. So a couple of groups have been pushing this. So one is our work, which was just published yesterday, and also a group by Dan Landau and work led by Asaf Ziran and Rafi Schulman and others, which was published a few weeks ago. They've also taken this approach where they're looking at integrating this data all across the genome. So when you take this approach, there are two main challenges. Challenge one is to collect as many informative reads, so you can look at these target sites. And the second challenge is to manage background noise. And both of these groups have approached it in different ways, and I'll encourage you to look at both of these studies. But in particular, I want to focus on our study for obvious reasons. One of these is that it was just published yesterday, so we're very excited about it. And it was a large group of people involved in it, but in particular, this was led by Jonathan Wan and Katrin Heider, two very good PhD students in the lab that have really pushed this project forward. So our first challenge is to collect many reads. And when we were thinking of many reads, we were thinking, can we really go as high as millions of reads so that we can really push our sensitivity to strong levels? So this relates to the depth of sequencing and the number of mutations you're looking at. And this led us to think of this in this conceptual framework, that when we're plotting here the amount of material that you're analyzing. So you see here, this can vary from the standard blood tubes, which give you approximately 10 mils of plasma if you have 2 blood tubes, and that gives you a few thousand copies of the genome. And after you do a library preparation, for example, or hybrid capture, this may reduce, so you'll be in the region of a few hundreds or a few thousand copies of the genome. You can go to larger amounts by things like plasmapheresis, but that's difficult to do, or you can even work with smaller amounts. And here we're plotting the number of mutations. And most of the literature up until a few years ago was really just focusing on this space here, where you're looking at a few mils of plasma, looking at one or a handful of mutations. And we wanted to explore this space here by looking at hundreds or thousands of mutations. How can we go in terms of the number of informative reads? So first was developing a process to do that on the laboratory side. So again, we built a process for tumor exome sequencing, and we chose exome sequencing at the time for reasons of affordability mostly. And then from this, we generated a mutation list. And then we've developed a hybrid capture panel that was specific, not for every individual patient, but for every set of patients. And this served two purposes. One was to contain costs, and the other is that this allowed us to generate a lot of background noise, background data. So we took a set of patients, set of mutation lists, and we generated one hybrid capture, and this gave us information for the target patient, but also background information in the other patients where this was run together. And then we ran this panel as well on healthy individuals, so we have control data both from non-matched patients and from healthy individuals. And the data, if you look at individual loci, can look something like this, where you have thousands of curves, each one showing you the time course of the levels of this particular mutation over time. But you can see here that for each individual mutation, our sensitivity is limited really by the amount of material. So at most we can get either 0 reads, or if it's 1 read, it's already jumping up. You can collate these mutations together to look at clusters, maybe identify subclones. But what we really wanted to do was to pool all the data together to really get to the point of looking at lots of reads. Sorry, PowerPoint is acting up on me, and this is not what the figure is supposed to show. Hopefully I'll be able to get to the other figure later on. But this is to show you here the number of reads we actually, informative reads we actually got. So by collecting a blood tube of material, several mils of plasma, sequencing that across a patient-specific panel with thousands, hundreds, or thousands of mutations, we could get this number of informative reads, numbering in the hundreds of thousands, sometimes even in the millions. Apologies, again, this is not showing what's intended, but this is intending to show you here how the process we've developed to reduce the background noise. So we had to pass the noise through the samples through the sequencing data through several filters, where we first did read collapsing by UMIs. That reduces the background noise by an amount, but this is still not low enough. If you have 1 million sequencing reads, you really have to get the noise down to levels of below 1 part per million to get the most out of it. So we added in additional filters, filtering out noisy loci, or we could require to have a read in both the forward and the reverse read, but really got the most benefit when we combined this to getting us close to the 1 part per million. And then by adding an outlier suppression filter, we could really get to noise below 1 part per million. This figure here was intended to show you that the noise levels varied between different trinucleotide contexts and different base changes. So some base changes like C2T were much noisier, and we couldn't really push that below the parts per million, but there were sufficient areas where we could push the noise low enough so that we could be confident to an extent with that. In addition to the noise reduction, noise filtering, we wanted to also give some kind of signal enhancement where we could. We did that on the basis of 2 factors, either using tumor information, where we enhanced where the tumor signal is very high, or using the ctDNA fragment sizes on the basis of work in the past few years by others in the field, including our own, published a couple of years ago, where we showed that if you look at the short fragments of DNA, this can enhance for ctDNA levels. So with these error suppression steps and signal enhancements, we got to the point that here in this dilution curve, you can see here the decreasing levels of ctDNA in logs. And you can see here that we were able to detect 2 out of 3 cases where the diluted DNA was less than 1 part per million. This was 0.4 parts per million. And at 4 parts per million, we detected 2 out of 2 cases, and so on, also with a good quantitative fidelity here. So having applied this to a cohort of patients with early stage melanoma that were measured after surgery, and advanced stage melanoma where these were monitored over time, you can see here our detection, and you can see here that the detection, the ctDNA levels spanned from order of 10% of the DNA to only a few parts per million in some of these patients. And when the levels were few parts per million, this corresponded to actually detecting of less than 1 cell's worth of DNA from a cancer cell. So we were detecting that in our blood sample, there was less than 1 part per 100 of a genome from the cancer cell, and we could still pick this up by piling together all of these loci. We took this forward further to study this in different cohorts, in a breast cancer cohort through our work with Carlos Caldas, in brain tumors, the work by Florent Moulier and Richard Mayer, and renal cancer, which turns out to be particularly challenging, as you can see here in the cases undetected, in work by Chris Smith and Grant Stewart in Cambridge, and in other cancers, including early stage breast cancer, which I'll get to in a moment. And you can see here the spread of levels of ctDNA, including in some cases at a few parts per million. One of the other nice features I like about this approach is that for each sample, you know how many informative reads you had. So even when you don't detect ctDNA, we know what was our sensitivity. And sometimes we have millions of reads, so we know we really had very good sensitivity, and we would tend to detect these cases even at very low levels. But in other cases, we ended up, because there were either a few mutations for this cancer, or we had very little amount of DNA, or the sequencing didn't work well. So we have only a low number of informative reads. And in those cases, at least we know that our sensitivity is not very good, and we can maybe correct for that or repeat, etc. So overall, looking at the cases where ctDNA was detected, we could see that in 10% of the cases, this was only in few parts per million. In almost 50% of the cases, this was less than 100 parts per million. And if you look at the cases we did not detect, we could at least evaluate and say that in 25% of the cases we could not detect, but we know this is no more than 10 parts per million. And in 75% of the cases, we could say this is no more than 100 parts per million. So having kind of gotten used to this framework where we look at the number of genomes we analyze and the number of mutations, we realized that although we can get to the millions of reads by looking at mils of plasma multiplied by thousands of mutations, we can also get to thousands of informative reads from very, very small amounts of information. And so we realized that by even with shallow whole genome sequencing data, meaning we need even less than a genome of one genome equivalent of information, if we have 1,000 mutations, we can get to sensitivities of parts per thousands or so. And this dovetails very nicely with other work we've done in parallel, led by Katrin Heider, showing that by analyzing that we could actually detect ctDNA in individual dried blood spots of DNA. So this is where a patient can do a pinprick, dry the blood, and then we can extract that DNA from filter paper and then detect ctDNA in those samples. Although some of that work is still to be demonstrated and developed, but we showed proof of concept how we can do that by taking blood and spotting it on cards. So you can see here some of our data from monitoring, and this is monitoring data, monitoring blood, looking at shallow whole genome sequencing data. And with shallow whole genome sequencing data, less than 1x, we can see that we can detect down to parts per thousand. And when we don't detect, again, we can at least cap and know that it's no more than some given number. And by simulating this, estimating the number of mutations that are in different cancer types, we can estimate what could be the level of sensitivity of analyzing ctDNA in blood spots in the future. So in closing, I just want to share with you some data that's really very much hot off the press, and we're going to share that in the ACR through a set of posters we're showing just next week. And this is data that emerges from our LUCID study, our lung cancer circulating tumor DNA study, where from starting in 2014, and this study continued in 2019, we collected plasma samples from 100 patients with non-small cell lung cancer in stage 1, 2, 3. You can see here the distribution. We had 60 patients with stage 1, 20 patients with stage 2, and 20 patients with stage 3, different histological subtypes. Now, I'm here to acknowledge and thank Robert Rintoul, who's the PI at the Papworth, Susie Harden was the PI at Addenbrooke's, Doris Russell, who was a lead pathologist, and again, a key help from Addenbrooke's Hospital, the Cambridge Clinical Trials Unit, and the Royal Papworth Hospital. And by applying our INVAR method on these samples, we got the following data. So of the 100 patients, for 90 of them, we had tissue available on which we could do exome sequencing, develop our patient-specific panels, and study it with INVAR. And you can see here the detection rates. We detected ctDNA in the majority of cases. And I can just say that for the first time, we were able to really define the median levels in stage 1 non-small cell lung cancer. And we find the median ctDNA fraction in stage 1 non-small cell lung cancer to be 12 parts per million. Importantly, we also detected almost 50% of cases with stage 1 adenocarcinomas. And in another study where we used these samples, we collaborated again with Inivata, and we used an adaptation of this personalized assay, which they call the radar assay. And in the radar assay, we use the Envision platform, this amplicon-based sequencing, but then we apply it on a personalized way by developing a patient-specific primer panel for each patient, undergoing, again, a modified workflow for Envision and sequencing this at high depth of 100,000x or so. And in this dilution series, you can see the sensitivity levels that we could get with this assay. Looking here at input of 20,000 copies, which would be 2 tubes of blood or so, you see that we could detect 20 parts per million at about 90% and even 70% or so at 10 parts per million. And this drops when we have less input, but it's still quite interestingly sensitive. And then we applied this assay, lastly, to a set of patient samples that were collected post-surgery or post-treatment for these patients on the LUCID study. And you can see here some examples. For example, a patient detected pre-surgery. After surgery, it's not detected, but then it's picked up a really few parts per million here and growing with a lead time of more than half a year before clinical relapse. This other patient here, which was even detected after surgery at few parts per million, was it 20 perhaps, and then a year later was detected with relapse through imaging and other effects. So again, in the posters presented at ACR, you'll see kind of the spread and these time courses followed individual patients, and importantly, this preliminary Kaplan-Meier curve. A lot of this data is still being cleaned up, I should warn, but we're very pleased with the results so far, showing here that the patients who have ctDNA detected have, as you can see, a much, much higher risk of early relapse compared to these other patients. So to summarize this talk, I hope it was interesting for you to hear me telling you about how ctDNA can help guide cancer treatment at different stages of the disease. My excitement about methods for targeted sequencing and how I feel that these have really allowed a lot of possibilities in ctDNA analysis. How we were using ctDNA detection at high sensitivity, why this is critical for both accurate molecular profiling and both the sensitivities that it can achieve in monitoring by using personalized assays. And I'm very pleased to see how the field is rapidly progressing to provide increasingly accurate data also to guide treatment in early stage cancer. This is very exciting advances. Last but not least, just my many acknowledgements primarily to the great team and the people who've been in the group and my alumni over the years, many of which I've mentioned through the talk, and others who have not yet listed here. I'd like to thank my funders, particularly Cancer Research UK and the ERC in recent years. I'd like to thank the University of Cambridge, which houses our group, Inivata, for all our work together. I'd like to thank our collaborators, key ones I've mentioned through the talk. And thank you very much. Thank you to the ECR and the Piscolar Foundation for the award today. And I'd be happy to take questions online if this works. Thank you.
Thank you, Nitzan, for this very exciting talk. And congratulations again for being the awardees of the Piscolar Foundation EACR Cancer Research Award of this year. I see you have received a few questions that I'll read out to you and that we can discuss live. So the first question relates to when we have ctDNA from different tumors in the same patient, do you differentiate? How do you know that it could a tumor in the lung or in the liver, for example? Basically, Ventura Garcia is asking you what happens if 2 tumors with similar mutation occur in the same patient.
Thank you, Alberto, and thank you, everyone, and good morning. So I'm not entirely sure I understand fully the question, but if you have 2 different tumors, obviously, if you've previously characterized the tumors, then you will be able to differentiate them by the different mutations. If they happen to have exactly the same mutation, then there is no easy way to identify a particular molecule. You can use things like methylation, but having a methylation pattern that's on the same fragment of DNA that you see the mutation on is probably fairly unlikely to be able to get that information. If you don't have information from the prior tumor, then you can work out various analytical ways to try to devise some information about that, but I don't suppose that would necessarily be easy or very accurate.
Thank you, Nitzan. There is an exciting question from Ludovic Barrault. He's asking, there are tumors that release less than others. There are districts of the body that release less than others, for example, the brain. But also, sometimes we have also observed this, there are non-shedders or tumors that shed less than others among the same tumor type, for example, in colon and lung. Do you have any clues as to why some patients will release more than others?
So there's various pieces of information that support reasons that could lead to that. One of the pieces of evidence, for example, that we and others have seen is that the growth rates of the tumor seem to play an important role, not only the tumor size itself, but the particular growth rate. So that could vary between tumors, could also vary across the tumor's life. That is one thing which relates to the biology, which there is evidence to support. Other relate more to the anatomy, if you will, of the tumor, where it's located, how much it's vascularized. But in a large part, this is still, remains to be explored and understood. But we do see, as you mentioned, quite a big variability in the amount of shedding of DNA.
Okay, then there is a question that is related to the previous one, but also ask you whether you have been looking at other bodily fluids, for example, urine.
Yes, we have looked at urine in different cancer types. For example, we've looked at urine in cancer types such as bladder and kidney cancer, where it's maybe more obvious to ask that question. And in bladder cancer, we found that it's very easy to detect, of course. In renal cancer, we found that it's difficult to detect, but we could still do that with sensitive methods such as INVAR. Interestingly, we've also been able to identify circulating or DNA coming from brain tumors in the urine of some patients using the very sensitive methods we now have for analysis. Generally, we see that the levels in urine are lower than the levels in the plasma, unless you have cancer such as bladder cancer. It's a possible source to look for information. The methods need to be adapted, need to be a little bit different, and you need to take into consideration the generally lower levels, and, for example, having shorter fragments, so the methods need to be able to work with that.
Thank you. Nitzan, I have a question myself as an estimator of you and the field in general. There is an entire strategy that has been formed to associate a mutation to a tissue, right? And that's possibly through epigenetics, but also through the use of multi-analytes, not only ctDNA, but other markers that can be either tumor specific or tumor associated. What's your take on that? Are you planning to include that in your studies?
Yeah, I think for questions such as early detection, I think this is important to try to get as multiple strands of information. We've seen, for example, work by the Johns Hopkins, including work they presented just very recently, a few weeks ago, where from their cancer-seq assay, where they combine information from ctDNA and from protein markers, they could detect some cancers through either. I think there are other markers that would be important to include. For example, methylation looks very important for early detection. So I think when one of these methods reaches its its glass ceiling, then it's important to include others. There's a question of what's practical to do, and if it's more practical to have multiple assays done in parallel or to have one assay done at greater depth. And those are questions that need to be bottomed out and explored. But definitely there's a role for multiple analytes to be studied together.
Thank you, Nitzan. A very important question about clonal hematopoiesis and how that could interfere. How do you take that into your calculation and analysis?
Yeah, I think that's emerging as a very important consideration to be looked at. A lot of interesting work by Pedram Razavi, for example, and others from Memorial Sloan Kettering, where they've provided a lot of evidence to show where that's the case. So I think we definitely need to consider that, especially if you're using things like gene panel assays, especially if you're using those for early detection, where you don't know the context of the cancer, then the likelihood of a mutation event that you detect in blood coming from clonal hematopoiesis could be higher. It depends on which mutations you're looking at, which genes. So all of these need to be put together, but it does suggest that in many cases, you would want to check in the buffy coat material as well, for example, and that needs to be done with at least a similar sensitivity than you're doing in the plasma. So I think it's important, depending on the application, that you sequence the buffy coat in parallel to the plasma. If you, with the exception of cases where you're looking at mutations that you previously characterized in the tumor, then you're in a bit safer hands with just looking in the plasma. But if you're looking at applications such as profiling or early detection, it's important that you also analyze the buffy coat.
Okay, I see more questions coming. I apologize to Caroline Dive, but I think we should address those that are coming from many other participants. So one that I think is very important is the economic of it. I mean, I see that there are connected people from all over the world, and some countries perhaps cannot afford some of these assays. Can you comment on the implementation in the economics of ctDNA analysis broad scale?
I think there are now a range of different methods that can be used to study ctDNA. Some are newer or broader or more expensive and would then be more expensive than others. Some methods can now be reasonably at lower cost if they use things like PCR-based methods, which require less instrumentation and lower costs in terms of consumables. The sequencing-based costs, some of them which require less sequencing, either looking at lower depth or smaller gene panels, could thereby reduce costs and get to a point where if you scale it up and do a lot of samples together, the sequencing costs can come down. Of course, you have at the other end methods such as the ones we're looking at now for very high sensitivity, which these are still very expensive because you need to sequence quite deeply in the plasma as well as get sequencing information from the tumor. These are still very expensive and at the moment probably suitable for research purposes and trial usage. What we hope is that over time, as there is greater and wider usage of this, then we will see a pattern, for example, like have seen with NIPT, where initially it was very expensive, but as the usage goes to the hundreds of thousands and millions of samples, then that helps scale down the costs because of efficiencies.
Okay, and with that, I'd like to thank Dr. Rosenfeld for this masterful presentation. Congratulations again for your award.